we're going to get going and talk about eukaryotic transcription regulation, specifically the GAL4 pathway. So up until this point, we've been talking about how bacteria sense nutrients that they have available and how bacteria regulate the expression of their genes that they use to metabolize sugars. We eukaryotes have to do the same thing. So last class we talked about the idea that there's an upstream activating sequence. And there could be a gene, for example, GAL1, that's involved in producing an enzyme that's necessary for an organism to use galactose, the sugar. So this is all about galactose utilization. I'm glad you're all excited it's Friday too, so am I. I know you can't tell, but I am. So we have promoter proximal elements, PPEs. That's where, for example, RNA polymerase is recruited to start transcription of, say, the GAL1 gene, which is an essential gene for chewing up sugar to use as an energy source. What was a UAS?
Upstream activation sequence.
So this is, UAS stands for upstream activating sequence. What does it do? What binds to a UAS?
The protein that you're going.
So a protein binds to an upstream activating sequence. In this case, for this specific case, for galactose utilization, that protein that binds to the UAS is called GAL4. So there are a number of genes that start with GAL. They all encode proteins that are important for using galactose. Right. GAL1, the gene here on the right, is going to produce a protein that's essential for digesting galactose. What is GAL4 going to do you think? What does it look like it's doing here in the way I've drawn it? How does GAL4 work to regulate the expression of the GAL1 gene? What could it be doing? What are the possibilities? Drawn like this, could it be repressing something? Is this acting like lac repressor? Just based on what's drawn up there. How does this differ from the lac repressor situation? How does lac repressor prevent transcription of the lactobaron? What is it physically doing? Yes, blocking RNA polymerase. Is GAL4 blocking the RNA polymerase? No, it's on the wrong side of RNA polymerase. It's upstream. It's in the 5' direction. So it's not repressing. What might GAL4 be doing in this case? So it's GAL4 is bound to the UAS, and it's actually positively interacting with RNA polymerase, like many other proteins we've talked about, sigma factors, TATA binding protein, all of these proteins that physically bind together to provide a base, essentially, for RNA polymerase to grab onto and be held onto the DNA to start transcription. So GAL4, to produce transcription of GAL1, you have to have GAL4 bound to upstream activating sequences. The problem is that GAL4 has an inhibitor. Its name is GAL80. Don't worry, we're not going to go through what all 80 of the GAL proteins do. We're just going to touch on a couple. Bless you. So GAL80 binds to GAL4 right there where GAL4 would normally be positively interacting with RNA polymerase. In other words, redrawing that down here on the bottom, we've got a promoter proximal element. Nothing is bound to the UAS. We've got GAL4 floating around here. Normally, GAL80 is physically bound to GAL4. And while Gal80 is present, Gal4 can't interact physically with RNA polymerase. So what do we want to do if we want to get transcription to happen? Which protein do we have to get rid of? Gal80. So we get rid of Gal80, then Gal4 should be able to come over here and actually physically interact and bring RNA polymerase to the promoter. So how do we get Gal80 off of Gal4? Does anybody know? It involves just one more protein you need to know about. Anybody? Gal1, Gal4, Gal80. There's a protein, I guess I'll use the third color, Gal3, that can get rid of Gal80. Under what circumstances does a cell want to transcribe Gal1? What environmental condition is the cell going to be in when it wants to produce Gal1 protein? Think about bacteria in the lactose operon. In what case does this cell want to produce a gene that will let it digest galactose? Yeah, sure, absolutely, when galactose is present. So how do you think galactose is going to affect these proteins? When galactose is present, which protein are we going to get rid of? Gal80. So when galactose is present, it positively interacts with Gal3. So galactose and Gal3 work together. The small molecule, the sugar, galactose, binds to Gal3. That's when Gal3 can remove Gal80 from Gal4. Right? Series of events, right? Cascade of events. And what happens when galactose is present and it and Gal3 have gotten rid of Gal80? At this step, what happens? You're right. Yeah, Gal4 can come recruit RNA polymerase and initiate transcription of Gal1. So in the presence of galactose, this is how we get transcription of the Gal genes that allow this cell to chew up galactose and use it to produce energy. Any questions? Now, if you're still finishing the evaluation, go for it. So I'm going to record this in case some of you happen to be distracted. Somebody last class asked about whether or not this has anything to do with lactose intolerance. And yes, it does. So everything we've been studying is directly relevant to those of us that can or cannot enjoy cheese, yogurt, other dairy products. The key molecule in lactose tolerance, you enjoy that stuff, right?
Yeah, I love it.
There you go, is the enzyme lactase. Right, so that's the protein that breaks down the sugar that's found in cow milk, any mammal milk for that matter, which is galactose and glucose together, breaks that bond. So it takes a complex sugar, this enzyme, and breaks it down into two simple sugars that our bodies can digest. But without this enzyme, lactase, there are problems. Well, they're socially embarrassing and maybe sometimes physically problematic, but you get wind, as it shows in this figure, if you're lactose intolerant and you eat dairy products. And sometimes wind is not something that you want. So what we're here to figure out is, and to learn about is, how does gene expression regulation have to do with whether or not you're lactose tolerant or intolerant? This is the normal situation, actually, in humans. So lactose tolerance is kind of a mutation. Those of us that can do it are sort of not wild type. And that's because normally human babies express lots and lots and lots and lots of lactase, the enzyme, because that's their diet is milk. And if they couldn't use the sugar that's in the milk, then the human species would not do very well. Right? So infants have lots of gene expression of lactase. And then at some point during most of our lives, not my life, but most of our lives, that lactase gene gets turned off. The expression goes down. So we no longer make it, then we become lactose intolerant. That's normal for most humans. But then we have the occasional mutants that continue to express lactose. They act like they're babies even when they're adults. So if you're lactose tolerant, that means that your gut is still infant-like in that it's still producing the lactase enzyme that lets you digest lactose, the sugar. So what's happening to make adult babies? Right? What mutations allow some adults to make lactase and some not? It's all about gene expression regulation, whether or not the lactase gene is being transcribed or not into adulthood. So it's a short story, actually. It's really pretty straightforward. Scientists started looking around at the DNA around the lactase gene, upstream, downstream, looking for what? DNA regulatory sequences. So we know that DNA sequences are important for transcription factors and enhancers, right? Are types of transcription, or types of DNA sequences transcription factors combine to and turn on gene expression. So geneticists started looking for, okay, where are the enhancers, where are these specific DNA sequences around the lactase gene that might involve it being turned on or turned off? And after looking at a lot of people that were either lactose tolerant or intolerant, they looked at their DNA sequences. In this case, they're looking, here's +1, you know +1 nucleotide, right? What's that? The +1 nucleotide is where transcription starts. That's the transcription start site of lactase. So just to orient you in these graphs, +1, that's where the lactase gene starts, and it goes towards the right. These scientists looked at a number of individuals here, represented by this top chromosome, individuals that have the X there. They don't produce lactase into adulthood. So these are lactose intolerant individuals. And they compared their DNA sequences, as you would hopefully predict, to the DNA sequences of individuals in the promoter region up here that do produce lactase into adulthood. They want to know, is there a simple difference? What's the genetic difference between people that can produce lactase as adult and those that can't? And they found there were 2 nucleotide differences shown here. That's it. So if you look at a bunch of people, there are random mutations that distinguish all of us from each other in this region of the genome. But when people were lactose tolerant, down here on the bottom, for example, there was always a T at position minus 13,910. What did people that were lactose intolerant have at that position? What nucleotide? The C. So there's a single nucleotide, this is a correlation, there's a single nucleotide difference that these scientists are basically saying they could diagnose whether or not you're going to be lactose tolerant or intolerant based on what nucleotide you have at that spot on your chromosome. What does it mean that it's at position minus 13,910? Why do I point that out? Because I'm lame. Yeah. Why is that interesting or relevant? Single nucleotide. At least partly caused by a single nucleotide change. How far away from the gene is it? In terms of how many base pairs away from the lactase gene is this mutation? 13,910 nucleotides. It's over 10,000 nucleotides away from the actual gene that encodes the enzyme that does lactose breakdown. In fact, what does this say? This mutation occurs in an intron in a different gene. It's that far away. On the chromosome near lactase, there's another gene called MCM6. And in one of its introns is this position minus 13,910. It's 13,000 base pairs upstream of lactase. So introns sometimes do useful things for us. This intron doesn't even have to do with lactase or lactose tolerance. It's in a gene that does something totally different. So that mutation then is called, in the literature, if you want to Google this, you can. I know you don't want to, but it's called the 13,910T SNP, right? The 13,910 comes from its position. And what was the T? That was a nucleotide. That was the nucleotide of people that were lactose tolerant. And what was the nucleotide for intolerance? It was a C. So if you got a T there at position minus 13,910, you were tolerant. If you had a C, you were lactose intolerant. So how in the world could a single mutation that's not even near the gene that these scientists are telling us it regulates, how could that single mutation have any effect on whether or not lactase is produced? One nucleotide change. At what point during transcription is DNA sequence important? The regulation of transcription. How could changing a DNA sequence cause a problem with transcription? Although very briefly, talked about a mutation in bacteria when you're talking about the lac operon. There's a mutation to a DNA sequence we could make that would change transcription of the lac operon. What was that? It's a particular class of mutations. So what was that DNA sequence called that was mutated that causes a change in lactose expression in bacteria? Lactase. What sequence does lac repressor bind? What is it? The operator. Hello, operator. Thank you very much. So if you mutate, in bacteria, you should remember this because this is on the final. If you mutate the sequence lac repressor binds to, what does lac repressor protein do? It doesn't bind. Right? Lac repressor binds a specific DNA sequence. If that DNA sequence isn't there, you get no repression. Here's a single nucleotide change. What's it going to do to gene expression? Here is the change itself. Right, there's a C, if you really zoom in, there's a thing there that says C/T. That's the position of this mutation. C, you're intolerant, T, you're tolerant. That happens to be part of a sequence that a protein called OCT1 recognizes. And OCT1 recognizes a particular DNA sequence, and this mutation happens to be in the part of the chromosome where that protein, which is a transcription factor, binds. In other words, this mutation could be occurring, this minus 13,910 mutation could be occurring in an enhancer sequence for that gene that's 13,000 base pairs that way, lactase. So a mutation really far away from the gene has an effect if, for example, that mutation either makes an enhancer sequence or abolishes, gets rid of a DNA sequence that a transcription factor would normally bind to. And in this case, that mutation that allows tolerance, which means you're the adult baby, you make lactase well into adulthood, people that have that tolerant SNP, single nucleotide change, the T, that improves the ability of the OCT1 transcription factor to bind to that site. What does that mean? It means that OCT1 protein is better able to keep RNA polymerase coming to the lactase gene and transcribing that gene and producing the enzyme that lets you digest dairy. So there's basically two versions of the binding site. Tolerant people have one type, intolerant people have the other. It's just a single base pair change. And it all has to do with how well this protein OCT1 can bind to that enhancer sequence and cause transcription of lactase. Questions or concerns at this point? This is a graphic of the GAL4 system, but we could replace this with terms we just used. So instead of this UAS, the upstream activating sequence, we now know that this was for which protein? What was the transcription factor that was binding minus 14, 9, 10? That was, this is an enhancer. What protein binds there? In this case, it's GAL4. In lactose tolerance, what's happening in my gut? Which protein is binding that enhancer for lactase? OCTN1. So if we want to make this customized for the story I just told, you replace GAL4 with OCTN1. Like GAL4, it's a transcription factor. It binds to an enhancer sequence. And instead of this example, then instead of GAL genes, that's regulating the gene lactase. But the process is exactly the same. When OCTN1 is bound to its DNA sequence, it brings RNA polymerase, right here, to the TADA box, right upstream of the transcription start site, and that allows transcription to proceed. So if this process is happening, what nucleotide is present here in the UAS? Is it the T or the C? When OCTN1 is binding there, bringing in polymerase, transcribing lactase. Is it the T or the C? It's the T, right? 13, 9, 10, T. So in this drawing, there's a T there in the UAS. If you happen to be the person or from a lineage of ancestors that has the C instead, none of this happens. OCTN1 doesn't bind to that sequence if there's a C there, therefore it doesn't bring RNA polymerase in, therefore you don't transcribe lactase. Therefore you can't digest lactase and you have the wind. And other issues. So that's the summary of how genetics impacts gene expression in a large number of humans. Yeah.
So how is it relevant, how is the enhancer relevant over time? So as you get older, how does that become relevant?
Oh, that's a fantastic question. So what happens as you age when normally as a baby you make lots of lactase? What's happening in the same individual who has the same DNA sequence is the essence of the question. Right? So it's not like as we age, we get mutations in this gene that automatically turn off lactase expression. Anybody have any thoughts? Over an individual's lifetime as they age, how is it that lactase gets turned off? I could play a chicken and the egg answer. I could say it's simply that there's less OCTN1 made as you get older. So you have less of this transcription factor, you get less promotion of transcription. But then you would have to ask me what?
What
Yeah, exactly. So then, okay, so you just pass the question on, well, what makes OCTN1 turn down as you get older? And then I have to say, I don't know. But that is absolutely the question. There's another thing that can cause changes in gene expression in your lifetime, and that has to do with this concept of epigenetics. You might have heard of anecdotally or in some classes before. We've got a whole week, next week or maybe the week after, devoted entirely to that question. So how does gene expression within an individual over time change? We've got a number of interesting cases to look at there. And so you might get your answer there. Yeah. But yeah, it's the perfect question to ask at this point in time. Since it's Friday, I figured maybe I wouldn't talk about pure genetics forever. It's from a genetics paper, but this is a map on top of where lactase persistence exists in the world. And so the heat map on the right there, you can see the reds, the hotter colors. 90% of individuals or so in those red colors have lactase persistence. So you can see where in the world most people are lactose tolerant versus areas of the world where people are lactose intolerant. And on the bottom, you get the correlation when you genotype people from different parts of the world, how many of them have the T allele. So these are people we would predict in the hot colors on the bottom map. We would predict that people that have, populations have 90% or more individuals have the T, we would say they're lactose tolerant populations. Okay. Do you see any interesting similarities or differences between these two? We've got the phenotype on the top, where lactose tolerant people are, and we've got the genotype on the bottom, where this mutation exists in the world. Are there any correlations? Where is there overlap? Yeah, in like Northern Europe, right, there's high frequency of this particular mutation we were just talking about, and that correlates well with high frequencies of lactose tolerance. What's more interesting than that on this map?
The fact that people in West Africa don't have the T in it.
Sure, so people in there's a really high percentage of people that live in West Africa that are lactose tolerant, but they absolutely don't have this one mutation. So what does that tell us about lactose tolerance?
That there's another gene involved.
Yeah, it means that there are other ways to become lactose tolerant into adulthood other than this one mutation at minus 13, 9, 10. So there are other genetic mutations you could have that let you be lactose tolerant. The same is true here in the Middle East, where there's a concentration of people that have lactose tolerance into adulthood. No correlation at all with this one mutation. So this mutation that we've been talking about seems to be specific to Northern Europeans. Which I am, and I had a friend about a decade ago that started a genetic testing company, and right when they launched, they needed people to be like their guinea pigs to make sure that their process was working correctly. So I volunteered, and they sent me a genetic testing kit, and I sent them some DNA, and they said I could pick 3 genetic tests I wanted to have done. And I knew I was lactose tolerant, so I figured it was a good positive control, because being a scientist, I thought I should do some control experiments. And it told me I was lactose persistent. And when I looked at my genotype, guess what my genotype at this allele is? So here we're talking about the LCT gene. It says I am a TT type. So both of my chromosomes, the one I got from my mom and my dad, both have a T. So of course I'm lactose